Welcome, everyone. I'm excited for everyone to hear this update. David Corey and Marina Ivanchenko have been an incredible gift to our community with their research progress. Um, so I am not going to talk long. I am going to turn it over to David, who will give his presentation. Please hold any questions until after. We have time for questions afterwards. Also, please keep your microphones muted during the presentation. Thank you so much. And now I'm going to turn it over to Dr. David Corey.
Hello, it's a pleasure to speak with you all this morning, or whatever time zone you're in. I should mention that I'm a professor of neurobiology at Harvard Medical School, and I've worked closely with Marina Ivanchenko, who's an instructor in my laboratory, and who's leading this project. I know that some of you are new to this and not everybody's kept up. So rather than give a progress report of the last year, I thought it would be useful to kind of give a global summary of the work that we've done to date and then where it's going. And this will involve some detailed science, but I'll try to give you the big picture of the study at the beginning and the end. And you can sleep through the middle if you want. So I'll start by sharing a presentation. And I hope that you can all see this. Yes, can we see it? Yeah, all right. So just to start with some of the basics, as you all know, Usher 1F patients are profoundly deaf at birth, and that limits our hope for a therapy that could restore hearing. I should say that they also lack balance, but it's possible that at some point the balance could be preserved. On the other hand, as you know, the loss of vision is slowly progressive, and that means that we have years, even decades, to possibly get a therapy into the retina. Now, the mouse models that we have for Usher 1F also show profound hearing deficit, but they really don't show much at all vision deficit. And so our strategy, as we've gone through this, is to try to test gene therapies in the mouse inner ear first, where there's a very strong phenotype. We can see the prevention of the deficit very early. And then if those strategies work in the mouse ear, we could then try to get them to the retinas of a number of model animals that we have. So as I said, we'll try to get to some other animals, and I'll show you some data with fish and with non-human primates that can give supporting data to the idea that some of these therapies could work for the retina. So just to remind you, protocatechurin 15, the protein that is defective in Usher 1F, is in two places in the body. One is in the photoreceptors of the retina. And I think you've all seen pictures like this. So if we take a little bit of the retina, I'm going to give us a laser pointer. Little piece of the retina, you can see that there are a number of different layers, but one of the outermost layers has the photoreceptors. And if we zoom in on that, we can see the rod photoreceptors that are sensitive to dim light and the cone photoreceptors that are sensitive to brighter and colored light. And what we know is that the protocatechurin 15 is right in this zone between the inner part of a photoreceptor and the outer part of a photoreceptor. And it's associated with these little finger-like things that are called calyceal processes. We can use an antibody to ask where the protocatechurin 15 is in a primate retina. And here in green, you can see the protocatechurin 15. So here's the inner part of in this case, a cone photoreceptor, the outer part, and the green shows that it's right in this little region. Now, we don't know exactly what the protocatechurin 15 is doing in that region. It may be important in generating this outer part of the cell. It may be important simply in stabilizing it, but we don't really need to know what it does in order to try to develop a gene therapy. We have a much better idea of what protocatechurin 15 is doing in the inner ear. So I think most of you know that the receptor cells of the inner ear that sense sound are called hair cells because they have this bundle of cilia-like processes coming up from the top. So here's one cell with about 60 little cilia coming up. And these cells are in the inner ear, both in the hearing part of the inner ear and in the balance part
Mm.
of the inner ear. So where's the protocadherin 15 in there? If we were to zoom in this section to just look at two of these cilia, you could see that coming from the top of one cilium to the side of the next taller one is this very, very fine filament that you can barely see here that's called a tip link. And we know from the way that sound moves these cilia, which is shown here, that every time the cilia go back and forth, they tend to stay stuck together. But you could imagine for that little tip link, that little filament, that every time it moves to the right, it's going to be stretched a little bit. And every time it moves to the left, it's going to be relaxed. What we know now is that this little filament pulls directly on another protein that lets electricity into the cell. And so when it pulls, the voltage inside the cell changes, and that's a neural signal that can go to the brain. And we know also that this tip link is made of two proteins. One of them is cadherin 23, which is also a protein involved in Usher syndrome. And the lower part is our protein, protocadherin 15. So if you don't have protocadherin 15, you don't have a tip link, you can never pull on these little pores. The cell never has an electrical response, and you can't hear, and you also don't have a sense of balance. So what can we do about this? Just to remind you about recessive inheritance, it only takes one good copy of the gene for protocadherin 15 in your genome to have a functional retina and a functional inner ear. And if a child is unlucky enough to be born with a mutation in both copies, then the child will have Usher syndrome. So what can we do? The standard kind of gene therapy uses a tiny virus called adeno-associated virus that's pretty safe and is very good at carrying DNA into cells. Um, and so we can put the DNA that we want into AAV and then deliver the AAV either to the inner ear or to the eye. So you could imagine that if a child, again, is unlucky enough to have two bad copies, then we could put the DNA for a normal copy into the AAV, put the AAV into the retina where it can diffuse to the photoreceptors, or into the ear where it could diffuse to the hair cells. And then once in the nucleus, this DNA will produce the normal protein. The big challenge for protocadherin 15, and this is really what we've tried to deal with over the last few years, is that it's a big protein. It takes a lot of DNA to encode it. And the amount of DNA is more than one adeno-associated virus can accommodate. So the big challenge in all of this is to find ways of circumventing this length constraint. And I'll tell you about three of the ones that we've tried. One of them has to do with the fact that there's a single mutation, what's called R245X, that is often found in patients with Usher1F. And if you look at this one little C here out of the 6,000 or so letters of DNA, that's converted to a T, but the TGA is basically a stop sign so that the protein is made up to this point and then just stops, and the rest of the protein is never made, and so it's non-functional. So if we could turn that T back into a C, we might be able to cure the disease. Well, the way that you can change letters in the cell's own DNA is called gene editing. And there are a couple kinds, and we've tried one that's called base editing. This was invented by David Liu at Harvard, and he's a real leader in this field, and we worked with him on this project. So the idea would be that instead of putting the DNA for protocadherin 15 into the AAV, you put the DNA for a little protein that's called an editor, and then the editor DNA goes into the cell, and it makes this protein editor And the editor can find that one T out of the 6 billion letters of DNA that you have in your genome. And then part of it is called a deaminase, and that basically fixes it and converts it back to what it's supposed to be. So we tried this. First, we created a base editor that would specifically target this mutation. And then we wanted to make sure that it worked. So before we tried it in animals, we made a cell line that has the same mutation, and then we can just study it in a dish. And we tried different combinations and different variants of this editor until we found one that we liked. Then we needed an animal model that would also have the mutation. So we engineered a mouse line that had that mutation. Then we can put the editor into an AAV vector, deliver it to the mouse inner ear, and see whether that prevents the hearing loss. So we saw some prevention of the hearing loss in the mice. It wasn't very efficient. We're a little discouraged about this approach because a new editor would have to be developed, and that probably the FDA would have to approve a new editor for each different mutation in protocaterin 15. So even though R245X is common, this is not very efficient. And so for all these reasons, we're not pursuing base editing as a strategy, at least not at this time. The second strategy that we used involves taking this long DNA for protocaterin 15 and just cutting it in half and then putting each half into an AAV vector. And then you would inject the two different AAV vectors into the same retina or the same inner ear. And by engineering the DNA, we can kind of create some magic so that inside the cell, the two bits of DNA are put back together. And now we have the full length normal copy of the DNA, and that can make full length protocaterin 15. So we've tried this strategy. And here the basic idea is to split the DNA sequence, and now each half can fit in one AAV. All right, again, we need an animal model. I mentioned the animal model that we made in a mouse that has the R245X mutation. For these experiments, we made another one that deletes the protocaterin 15 gene only in the inner ear. And that also produces a deaf mice. And I'll tell you later about a zebrafish model and a primate model. So we engineered the dual AAV vectors, and then we also engineered this mouse model that lacks protocaterin 15. We injected the two AAV vectors into the mouse inner ear, and then we wanted to test whether it worked. And there were three assays for whether it was working. One was to see whether the protein that is made goes to the right place in the cells. And you can see here that it does go very well. So when we put the DNA for the AAV, for the protocaterin into the AAV, we put a little tag on it so that we could see where it went. And here's kind of a top view of about 9 hair cells, maybe 10. And you can see the little purple dots are where the protocaterin 15 is made from the AAV that we delivered. And it's going to the tips of the cilia, which is exactly where we want. And if we look at that with electron microscopy, these little yellow dots are where the new protocaterin 15 is localized. And you can see that it's exactly where the tip links are between a short stereocilium and a taller stereocilium. So the protein is made and it goes to the right place. So that's very promising. Next, we want to see, can it prevent the deafness in this mouse, and can it prevent the loss of balance? So here's one aspect of the phenotype in the mouse, and that is if we look again with the electron microscope at the bundles of cilia in a cochlear hair cell, they have this beautiful sort of V shape with several different rows of cilia. In the mouse mutant that we made, you can see within about 30 days, these cilia are starting to degenerate. And of course, they're not functional. If we take the mutant mouse and treat it with this dual AAV that is delivering the protocoherrin coding sequence, you can see that that prevents the degeneration of the bundles. So now we're hopeful that that will also allow the cells to function properly. Here's the way we test the hearing. And I'll just take a moment to explain this. So this is called an audiogram. And what we do is play a tone at a certain frequency, say 11 kilohertz. Mice can hear pretty high frequencies, up to 45 kilohertz or even more. And then we increase the sound level until we just see an electrical response that's measured from the skin at the back of the head. And then we'll call that threshold and say that this normal mouse has a threshold of 30 decibels. That's pretty quiet. The mutant mouse, you can turn up the sound and turn up the sound and turn up the sound and still get no response, even at 85 decibels. So that's a very deaf mouse. But if we introduce these dual AAVs into the mutant mouse, then the threshold goes back down to maybe 45 decibels. So that's getting pretty good. So each of the dim lines represents a different mouse. This solid line is the average of all of them, and the dashed line is the mouse that had the best response. So the very best one is getting hearing just as good as the normal mouse. So that's very encouraging. Now, I mentioned, of course, that the hair cells are in the vestibular part of the inner ear, the balance part, and the mice also show defects in balance. So one of the peculiar things about mice that have a vestibular defect, a balance defect, is that they tend to run in circles. So if you put a mouse into a box and watch where it runs around, it tends to hug the edges of the box. Maybe it's a little scared of the open areas. A mouse with an inner ear problem, with a balance problem, runs in little circles, and we can measure how many little circles it runs in. But if we take the mutant mouse and inject these dual AAVs, then it's not running in circles anymore and it's hugging the walls. And we can quantify these pictures with this plot here. So the normal mouse doesn't run in circles. The mutant mouse that we did does run in circles, but the mutant mouse that's treated with the dual AAV no longer seems to be dizzy and no longer runs in circles. Another thing you can ask is, can the mice swim? And a normal mouse can swim quite well and will swim for 60 seconds or even longer until we get tired of watching. The mutant mouse really can't swim, and it kind of tumbles and we pull it out of the water very quickly within a couple, maybe 20 seconds. But the mutant mouse that's been treated, once again, can swim for a long time. Or here's a third test. We can see whether the mouse can stay on top of a cylinder that's rotating. The normal mice can do it for more than 30 seconds. The mouse mutants cannot at all, and the treated mice can run on that. So this is pretty good evidence for restoration of the balance. And this is something that we might think about down the road in treating human Usher patients. What about the retina? Well, we don't have a primate model of Usher 1F, but we can ask whether the dual AAVs can carry the protocaterin 15 DNA into a non-human primate retina. And we did that, and we found that nearly 100% of both the rod and the cone photoreceptors received both AAVs and made the protocaterin 15 protein. So here it is shown in green, and you can see it in the calyceal processes, and you can also see it a little bit in the outer segment part of the photoreceptor. So nearly 100% is very encouraging. And then, well, a monkey isn't exactly the same as a human. So in collaboration with a group in Switzerland, we delivered the same AAVs to human photoreceptors that are grown in a dish. And we found that they also made the protocaterin 15. So we can't test whether it works in the primate photoreceptor, but at least it's being made by the cells and going to the right place. So I would say this is pretty encouraging. The third strategy that we've used is what's called a mini-gene strategy. And the idea is if the DNA for protocaterin 15 is too long, maybe we can just shorten it a little bit take out parts that are not absolutely essential, and then the shorter part of the DNA can fit into one AAV, and that can be delivered to the cells. And even though it's not full length, it might be able to make a functional protocadherin 15. So to figure out what part had to be cut out, we looked very carefully at the structure of the protocadherin 15 protein. And this is work that was started with Marco Sotomayor, who was a postdoc in my lab and now has his own lab in University of Chicago. Actually, I'll be talking with Marcos later this morning. And Marcos figured out where every one of the 16,000 or so atoms in protocadherin 15 is located and how they attach to each other. And what we saw was that this filamentous part of protocadherin 15 is sort of like a chain with 11 distinct links. And we know that this part of the chain has to bind to the cadherin 23. This part of the chain has to bind to the ion channel. But maybe we really don't need all the links in the middle. And maybe we can take out some of them to make the protein shorter. So we tried a number of different versions where we took out different amounts and finally settled on one of them. And here, I'll show you that we took the same mouse model that is completely deaf. And if we add a single AAV with the DNA for this mini protocadherin 15, then we can bring the hearing sensitivity of the mouse almost back to the normal mouse hearing. And so that is pretty encouraging in the mouse cochlea. Um, well, again, we'd like to be able to go to the retina. We need to show that this works not only in the inner ear, but in the retina. And as I mentioned, there's not a non-human primate model of Usher 1F. And we think to make one would take a long time. It would be expensive. And also the loss of vision might take years. It may be a very old animal before we would know whether it was really losing vision. But we do have a fish model of Usher 1F that was made by Monty Westerfield and Jen Phillips at University of Oregon. And it does show degeneration, and it does have reduction of the visual sensitivity, and that can be measured in a couple of ways. So here's one way of measuring the visual sensitivity, and that is, so this is the little fish. It's only 6 days old, and it has pretty big eyes relative to the rest of its body. And you can bring a little electrode down and just touch the surface of the eye and flash light on the eye, and then measure the electrical voltage that changes when the eye receives the light. So here is the result of flashes of dimmer and brighter light, and the electrical response gets bigger and bigger. But in the zebrafish that's missing protocadherin 15 that Madi made, you can see that the response isn't as big. So this electroretinogram has a smaller response. And we can measure two parts of the electroretinogram, a little one in the beginning that's called the A wave, and the larger one that's called the B wave. Another way of testing the visual function of the fish is to see whether it will follow patterns that are moving. So the way that we do this is to put the fish in a dish and then have a drum with stripes around it that moves in a circle. And then fish, and also we, would tend to track the stripes, and our eyes would move to track it. So here is an image showing how the eyes are moving as the stripes move around. And when they can't go any farther, they flip back and then they move more. And by analyzing the video frames, we can see how the left eye and the right eye move, and then we can quantify that. Does the delivery or the insertion of a mini protocadherin 15 into the genome prevent that? And the answer is yes. So if we measure this optokinetic reflex, the following of the visual stripes, the normal fish makes this number of movements in a certain amount of time. The mutant fish only makes about half of them, but the mutant fish that has been given the mini protocadherin 15 again makes the normal number. If we measure the electrical response, again, we see that the vision is rescued. So here is the amount of voltage that's generated by a flash of light, and it's smaller in the mutant fish, but the mutant fish with the mini protocaterin 15 has a normal amount of electrical response. So this is really good. It's showing that this mini version is functional in a retina, at least a zebrafish retina. Well, we'd still like to go into a primate. And even though there's not a primate model of Usher 1F, they are useful for determining whether the photoreceptors can make the protein, whether the protein goes to the right place, and importantly, whether the treatment causes toxicity. And so together with Spark Therapeutics, and many of you know Spark as the company that developed Luxturna, the treatment for LCA2, with Spark, we tested this in about 14 monkeys, injecting the mini protocaterin 15 encoded by an AAV into the primate eyes. And then for each of the retinas, we would take different parts of the retina. So the injection area is this light colored area, and we would map in different parts whether the cells produced the protocaterin protein and whether that spread to the areas outside the injection. And what you can see is that in an injected area, pretty much all the cells are making the protocaterin 15, and it's going to the right place between the inner part and the outer part of the photoreceptor. And if you count the number of cells that make it, between about 70 and 100% of the photoreceptors are making the mini protocaterin 15. So that's a pretty good delivery rate. As I said, it's located in the proper place. And then important issue, is it safe? Does it cause any toxicity? And we did see that the injected area caused some inflammation, but if we treated the animals at the same time with corticosteroids, that pretty much eliminated the toxicity. And that's a kind of a standard thing for gene therapy to treat with corticosteroids. So we think, based at least on these monkey experiments, that it's safe. Okay, where do we go from here? In general, we want to do a clinical trial. And for FDA approval of a clinical trial, we need to show that the treatment works, that it has efficacy, and that it's safe, that it doesn't have toxicity. So we've shown efficacy in the mouse inner ear and in the zebrafish retina. And based on that, I think we would get permission for a clinical trial. We've shown that it's safe, that there's low toxicity in the primate retina. We'd like to do a clinical trial, but that's difficult for an academic laboratory to do. Importantly, the application to the FDA for a clinical trial is a very complicated thing. It can be 1,000 pages or more. And so we're looking for a company at this point, first of all, that has experience in these applications to the FDA. We'd like to find a company that already has experience in gene therapy for vision. And we'd like to find a company that's willing to put 10 or $20 million into a clinical trial. At the moment, finding a company is not that easy. And the challenge is, as you all know, that Usher 1F is pretty rare. And companies are going to worry that if they put that money into a clinical trial, they're not going to make it back, that it may not be profitable. Nevertheless, we are talking with a number of different companies, and the hope at this point is that we'll find one that thinks that this will actually be profitable and that could be added perhaps to their existing portfolio of gene therapy for vision. So let me just end by mentioning that this is all the work of a large team that's taken place over between about 5 and 7 years. But I want to particularly mention Marina Ivanchenko in my lab, who's really led the project from almost the onset. Marina's trained both in the inner ear and in the retina. She's an ophthalmologist, and so she's really ideally suited to lead this project. And with that, I can take questions.
Okay, if you have a question, unmute yourself and ask the question. Okay, well, I can start. So one of the things I'm wondering about is we know that like all of our patients are at different stages of the disease. And I know you talked to me in the past about the cells first going dormant and then they die, and you think that you can reawaken the dormant ones. I know it was an anecdotal report, one patient who received the Usher 1B gene therapy out of Italy did report an improvement in vision. So I'm wondering if you can talk to that at all.
Well, I mean, it's a great question. I don't have a very expert answer, but I can speculate. So we don't know exactly what's happening to compromise the function of the photoreceptors. It seems that a protocaterin 15 is involved either in the production or the stabilization of the outer part, the outer segment of a photoreceptor. And so we might see in a primate that had the disease that the outer segments got smaller and smaller, and then the cell that might die. But we know that the outer segments are being continually um, renewed in a normal retina, and that is the outer segment may only last for a week or two, and new outer segment is continuously made. So if we were able to restore the function of protocaterin 15, it may be that the cells could regenerate their outer segment before they degenerated. So without any expert knowledge, I can say I would be hopeful that there might actually be restoration of vision.
Okay. Any other questions?
I have a question. This is David Creamer.
Go ahead.
Thanks. Hey, Dr. Corey, thank you very much, very, very much for the presentation. Really appreciate it. Is I noticed that you summed up by saying that you're looking for a company to take the mini gene through the FDA approval process. I'm curious why that versus the dual AAV. Is it based on the efficiency, the science, market, the ability to bring one to market versus another? Can you comment on that?
There are two, I mean, I would say that both are working, but there's a few reasons to favor the mini gene. One is that it involves production of only one AAV instead of two. And so in a sense, you can get twice the concentration in. In the mouse inner ear, it worked a little bit better to restore hearing than the dual AAV. So for those two reasons, I think it's just a little bit simpler and maybe a little more efficacious, but both are working. I should say also that we're now working on a second way of delivering dual AAVs that involves a different way of joining the two parts together. And in the mouse inner ear, that definitely works better than the first dual AAV method that we tried. So it's possible that this new method that we're playing around with now could work even better than the mini, but we don't know yet.
And do you have any thoughts? I don't know if this is your area of expertise at all on the ability of one versus another just to get through the process of FDA approval.
I would guess that they would be equally acceptable to the FDA. Certainly, the FDA has approved other dual AAV methods, and as long as we show that one or the other is safe, I think it would be fine with the FDA.
Great, thank you very much.
Any other questions?
I just want to, this is Jared Root. I just wanted to thank Dr. Corey for all your hard work over the years. And we know this is a complicated process, but we are the USH1F, I can probably speak by most, are very hopeful and positive. Is there any um I know that you probably also communicate with the Foundation Fighting Blindness, but I know that their pockets might be deep. Is there any interaction with them in terms of your process and maybe having them work together with us on this in terms of going to the FDA?
So two things. One is we are in touch with the Foundation Fighting Blindness, and they gave us a very nice grant to carry some of the work further into the zebrafish retina and into human retinal cells in a dish. And that we wouldn't have gotten that without the preliminary data that we could get with the support of the Usher 1F Collaborative. So that's been a really great partnership. In terms of going to the FDA, I'm not sure that the Foundation Fighting Blindness would help with going to the FDA, but they are helping us to find a company that might take it to the FDA. So that partnership is working out really well.
Thank you.
All right, last chance for questions.
This is Diana Sackett. I have, first of all, when we heard you talk and explain in the beginning of our journey, 8 years ago, I think it was in Chicago, Dr. Corey, it was just amazing that any of this was possible to talk about. And following this journey has been overwhelmingly just astonishing. I'm so grateful for such science. As we look forward to the hope that this could bring us as parents, because our son has Usher 1F, how can we support whatever it would need? Like, are there possible things that he could be doing to retain what he has retinal health wise, as long as possible to be ready for the gene therapy if and when it's available to him? And then my second question is, what can we do to try to help make this money come to fruition?
Yeah. So your first question, I think, is an important one. That is, while we're waiting for a cure, what can you do to preserve the vision? And this is really not my area. There are ophthalmologists who would have a better idea, but I think there are some promising drug strategies. There's the NAC that seems to be helpful. And I know that some Usher 1F patients are taking that with some preservation of vision. I would say just very simply, bright lights are not going to help. And so sunglasses, whenever the light is too bright out there, would just kind of an easy safety thing. The second question about helping, well, I should say that we wouldn't have been able to do any of this without the support of the Usher 1F Collaborative. And it's slow work, it's expensive work, and the fact that all of you have been patient and been willing to contribute has made this possible. We really, really appreciate it. If you learn of other foundations or people that would be willing to support it, then please introduce the collaborative to them, and that will help. Or if you know of companies that might want to pick this up, then give us introductions to them.
All right. And Diana, I just want to add that we have undertaken another project with a company called Unravel Biosciences that using an AI model tested 40,000 already FDA approved drugs for efficacy for Usher 1F. And we just got the report and I'm meeting with them this week. So I am going to be letting everyone know about it. I just need to understand the results myself to be able to let everyone know. The idea behind this project is that that would be the fastest path into patients because a drug that's already FDA approved wouldn't need safety testing and could be prescribed off label. And so our thinking in funding this was to buy us time while we're working to bring the gene therapy to clinical trial.
I had a quick question. That's Nick here, Dr. David Corey. Just want to say also thank you very much for what you're doing. Um, I just had a question regarding the clinical trials and the FDA approval. Once that is done, I guess I have two questions, but once that is done, is that pretty much it? Does it need to just go out on the market from there? And also, um, I guess this is for other countries as well, but how does it go? Maybe you don't know this, but how does it go to the other countries, like globally? Is that approved, like for Canada, for instance? Does it have to go through a different system? I don't know if you'd be able to answer that.
Yeah. So the path to clinical approval has several steps. First, you would meet with FDA representatives in what's called a pre-IND meeting. IND stands for Investigational New Drug. And you're basically asking the FDA, what would you like to see in order to give us permission for a clinical trial? And they'll tell you, and then you put together this big package that's 1,000 pages or more with all the data that you have, and you come back to them and say, here's our package, will you give us permission for a trial? And if you get the permission for a trial, then that could be a 2 to 4 year process where a dozen patients might be identified and treated, and their vision very carefully monitored to see whether it improves. And then if that's successful, the FDA would give you approval for the drug to be used in general clinically by any surgeon that's qualified. In terms of approval in other countries, I really don't know. I would guess that it would have to go through a similar approval process in a different country, or of course, a patient could come to the United States for treatment.
Thank you.
Okay, any other questions? If not, I'm going to turn it over to our wonderful development director, Sarah Gouch. So let me know if anybody has anything else. If not, I just want to say thank you so much. Dr. Corey, this has been wonderful for our families to hear, and you give us all hope.
Thanks.
Okay. Thank you, Melissa, and thank you, Dr. Corey. Every time I hear Dr. Corey speak or see one of his presentations, it's mind-blowing to me and amazing. And I think it's all really, really good news, that the work that's going on in your lab and others. I am going to share my screen now. Let's see here. Okay. Goodness, wait a minute. Are you seeing my screen yet?
We're not seeing
We're
it.
not seeing
Also,
it.
and there's one more question in the chat.
Okay. If that person would like to ask the question.
I can answer that while you're working on your screen. So Abby asked about since many of us are part of the clinical trials, will all of us be part of this once the FDA approves, or will a small number of us be selected? So typically in a clinical trial, a phase 1 trial is for safety. And so they like to be sure they're not going to cause any harm, which means that the patients initially selected will have lost more vision, not they want to be able to see some results. But once they determine safety in a few patients, then they open it up to more patients. A phase 1/2 clinical trial is also looking at efficacy. We are fortunate we have our natural history study so that we have data to compare. And the reason we invested in this was so that everybody would be able to get the treatment. This would not be a clinical trial where some people get a placebo or a fake drug. And now I'm going to turn it back over to Sarah. We are
Okay.
seeing your screen.
Okay, thank you. Great. Okay. So the like I said, it's amazing news that all the work that Dr. Corey and then all the other scientists in the collaborative have also undertaken over the last several years. I know it's daunting news that when you have a rare disease, it seems to be put on the shoulders of the families to fund the research. And that's just the way our world works, unfortunately. But you are so fortunate that years ago, the Chaykoffs founded this organization. And the reason that they did was they asked themselves the question, if not us, then who? And that's true for our entire organization and our collaborative. It's going to take the families to step up and work together to get us over that the roadblock and get us through the FDA funding or FDA approval. Um, our scientists cannot continue their research without the funding from Usher 1F Collaborative, and they do get other funding as well, but the Usher 1F Collaborative has been huge in funding Dr. Corey and our other scientists. How can our families help? There's so many very easy, very not time-consuming ways, and I know fundraising is also daunting, but everyone's community wants to help, and there's some really fun, easy ways to hold fundraising events. Every single event listed here is something that someone within our community has completed within the last year. We have a workout event, almost a model that Dory and the Roots and the Chaykoffs have already formed. And I'm going to let Dory talk about that in just a minute. Restaurant night, there's so many restaurants in every community that if you reach out to them, they'll donate a portion of proceeds from a certain night to a nonprofit. So you can reach out, share it with your community, come eat at this restaurant on Tuesday night, and they'll donate a portion to Usher 1F Collaborative. And kind of the same concept with the shopping event in local stores. And at the beginning of next year, we'll have the Super Bowl right away, followed by March Madness. I think everyone enjoys participating in some Super Bowl squares or some gambling with the March Madness. And that always is a good fundraiser as well for nonprofits or for Usher 1F specifically, or something as simple as a bake sale as well. Similar to what a lot of our families did for for Giving Tuesday, you can create and share a fundraising page, which we have the software for that, and we can work with you on that in honor of a birthday, an anniversary, a bar mitzvah, bat mitzvah, anything where someone would be giving gifts. What would be a better gift, right, than helping a treatment for you or your child? And then another really easy way is corporate matches. So many companies will match the donations made by their employees. And it's usually as easy as filling out a form on your company's portal. So check into that and check and see if you make a donation, if your company will match it. With that, I'm going to turn it over to Dory to talk about her most recent event called Sight Sound Sweat that raised money for Usher 1F.
Hi everyone. Thank you all so much. Again, Dr. Corey, thank you so much for your amazing presentation. It will give me a lot of enthusiasm knowing that we're all contributing to that in our own way. But yeah, everyone loves working out. It can be a fun dance class. It can be a fun spin class. It can be just anything you can think of, like obstacle course. You can even do a workout fundraiser for your young kid. It can be like a little gym and just something simple like that. And then pair it with what we all are here for, is a cause that's near and dear to our heart. Most of us are affected by it in some way. And so, the one thing is you gotta tie it with something that people love doing. And it's just an easy way to get people to be excited about it. It's literally probably an hour and a half at the most of someone's day. So it did not take the full day. And it's just something to take that into consideration. That's how you sell it to people. It's like, hey, this is literally an hour workout. You can try and get the local restaurant or maybe even some friends that will offer to bake or cook for the event. You can offer some free food, maybe a coffee bar. So that is what I did. Is I found a couple of people that were willing to sponsor a coffee bar. So we had a coffee bar, and then we did a workout in the morning, and so people were able to enjoy some coffee and some food. And that's another way to entice people to show up. Is hey, you fundraise an X number of amount of dollars in order to work out. and in return, you'll have a great workout and you'll get to meet people, you'll get some free food and some drinks, and you're all there for a great cause. For this workout, I asked everyone that you must raise at least $75. And then if you want to raise more than that, amazing. I also made it a little bit of a competition to say if you can raise the most money for your own workout, where you can, reach out to people in your community to say, hey, I'm doing this workout. I'm trying to reach my personal goal of $200. You can literally just ask 20 of your friends and family to donate $10 each. Easy, right there. And the way you can also sell it is to say, give up one coffee or drink that you normally would spend that day, and you can donate it to your friend or your family friend workout. So, there's a lot of easy way. I am available. Like, I love event planning. I love that kind of stuff. I personally will help you. You tell me who the gym is, if it's a gym that you go to often, or you have a family friend that is obsessed with working out, ask them, hey, would you help me ask your culture to put this on in your gym? Hey, if it works out, I will personally fly out and help you run the event. That is how important this is to me. So it should take a little bit of time and effort, but the results are amazing. A couple of years ago, I did it in San Francisco, and I have raised over almost $15,000. So, it should depend how many people you can fit in. There were over 50 people at this workout. So it was pretty incredible. If you have any question, Sarah, I think had provided my phone number and my email. You can text me, you can email me. I am available. I want to help you put on your event. Same thing with shopping event, anything you can think of. Even restaurant. I'm a foodie. I can help you find local restaurant. But yeah, thank you for letting me speak to that.
Thank you, Dory. And I want to point out on this slide too, if you look at these donation amount segments, again, I know fundraising can be really scary and daunting for some people, but the majority of the donations that came in on Dory's event were less than $100. And she ended up raising $7,465. It all adds up. So you don't have to ask people for a lot of money. Just show them your enthusiasm and how much it means to you. So this slide, just to leave you with a parting thought, if we had 20 Usher 1F families to each raise $5,000 in a year, and that will equal $100,000 of research that we can invest in our labs. And as Dr. Corey said before, the funding that Usher 1F Collaborative has given has opened up doors to even larger funding through large organizations like Foundation Fighting Blindness. And that wouldn't have happened without Usher 1F Collaborative support. If you need any help, here's Dory's and my contact information, our email addresses. Please reach out to us. We're very happy to help get you started and help out any way through the event. So I think that's it on my part.
We have one of the deaf participants raising their hand. Jamie. She's just going to pin me for 1 second. She has a question for somebody. Thank you for your patience. Okay, I can see better now. Thank you so much. I just have a question for Dr. Corey. It might be a little bit off the point, but I've been trying to raise my hand. I don't think anybody could see me, but thank you, Dr. Corey, so far for your amazing research and the teamwork that's happening. And I just want to get clarification for Usher's 1F, those of us who have it. We want to prevent our eyes from getting worse. We're supposed to use sunglasses outside. Is there other tips that we could use to preserve as much of our sight that we can?
Um, Yes, I'm not an ophthalmologist and I would not have the best advice, but I know that there are some strategies and perhaps some drugs that could slow the progression. I think Melissa is very aware of some of them, and maybe she's the best person to answer this.
Yeah, Jamie, I would say that you're an outstanding example of one strategy that has helped, which is they have shown that aerobic exercise uh, helps preserve the vision longer. I think it's related to oxidative stress. And for those who don't know, Jamie's a marathon runner, and she has run a couple marathons for us, including the Boston Marathon, and is unusual at her age to still have some usable vision. So that is one other strategy is aerobic exercise. So Jamie, keep running.
Okay. Um, now, so people who have ushers, you would suggest always use sunglasses. As a patient of Usher's 1F, have there been strategies for this one specifically, or anything else that it has been known to work?
So what I would say is stay tuned because we did just get the final report for this uh, testing drugs for efficacy. And so our next move is to identify probably about the top 10 drugs and start testing those in our zebrafish models. Once we have a full report on the drugs that could help you, we will be letting everyone know. And these, again, are drugs that have already been approved by the FDA for other purposes. And so we would be doing what's called repurposing them for Usher 1F. That is a very hot topic for rare diseases right now, is
Now,
repurposing existing drugs.
are they specifically testing patients that have Usher 1F? Or
Yes, yes. We actually collaborated with some Usher 1B families. So we've tested for both because we reasoned if we got hits for that would work for both, that we could share the cost of the subsequent testing.
and can you share that, who the patients are that have 1F with us, or is that private information?
No, I'm not allowed to HIPAA restrictions.
I see. I see. Okay. Thank you so much.
But I can tell you that it included um, a few different mutations to make sure that we were getting applicability for everyone with Usher 1F.
Also, the test itself, is it for older people, or are we looking at people who are completely blind at this point and to see if it works? Or are we doing little babies? What is the demographics for
You mean
people
for
that
the
are tested?
drugs? For the
Yeah,
drugs,
for the
it's
test.
solely based on the gene. So it would apply to everybody with Usher 1F.
Okay, thank you very much. Oh, and my last question is, if a patient themselves are fully blind, have no sight, would this kind of would this might be able to work for them to get some more sight back, or is it is once they're totally blind, it's too late?
The gene therapy or the drug therapy?
Either one.
Um, for the drug therapy, I would say probably not. However, I know that in testing a drug on our zebrafish that our University of Oregon lab did the testing, it's something called hexafluoro. They have published about that, and that has improved vision some. Um, and in terms of the gene therapy, again, you know, I think we talked about that. It's possible. We won't know for sure until a clinical trial.
Okay, thank you so much. That's all I have. Thank you.
And with that, we are at time. Just so everyone knows, we have recorded this, and so we will have it for others to watch again or to share if you would like. And I just want to say thank you again to Dr. Corey, and thank you, Kim, for your interpreting.
Thank you.
All right. Bye, everybody.